Well, cool. All right, so yeah, we'll get moving on with the presentations. So the next presenter is Maxwell Loang. He is a PhD. He is he is an assistant professor, geez, at ASU's New College of Mathematical and Natural Sciences. So he will be going over the environmental contaminants of cannabis sativa. So Max, if you do want, you already have your camera on, so that's great. So then if you do want to share your screen now and then get started with your presentation whenever you're ready.
Okay. All right. Can everybody hear me okay?
Um,
All
oh,
right.
Yes,
Okay. Yeah. Thank you. Thank you. All right. Okay. Well, thank you so much for the introduction, Travis. Great to see you here today. And also thanks for the invitation of the council for the opportunity to speak to you all with an interest in food safety and environmental health. Very excited to be here. So let me first introduce myself. My name is Max. I'm an assistant professor in the pharmacology and toxicology program at ASU. I also direct the ASU laboratory for cannabis sativa and One Health. So we are one of the two DEA Schedule 1 research lab in the whole ASU system. And we are very excited. I'm very excited to be here talking to you all today. So in the next 15 minutes or so, I would talk to you all about my research. First, I want to introduce, like cannabis sativa. We introduced cannabis sativa as a species and also what One Health is about. Then I want to talk a little bit more about our policy, our regulatory policy research on environmental contaminants in cannabis. What are they? How are they being regulated in the US? Now, next, I want to talk about environmental contaminant in the context of how does it affect patient and physician, in the healthcare system. The knowledge of, cannabis contamination. How do we introduce risk into patient and physician education? And I will also talk about some of the laboratory work that we are doing at the Schedule 1 lab, looking into using new approach methodology to predict combined toxicity of cannabinoid and pesticide. Last but not least, I will talk about the latest research direction, some of the questions that we are currently pursuing right now with regard to fungal and mycotoxin contaminant in cannabis. Now, first, let's talk about cannabis sativa. So, our lab are just research in cannabis and also industrial hemp, looking at both of them as an agricultural crop. Now, the reason why we are very interested in this particular species is that there are many different identities come with this one species, cannabis sativa. If you're growing it, uh, for textile, plastic, constructional material. Some of you may have heard about the term industrial hemp. So this is a term describing cannabis sativa that do not have high level of THC. You mostly are growing it for fiber. So that's one application. And also you can grow cannabis sativa for seed, and then that would be for protein food sources for human and animal. So that's another key application. Now the third use of cannabis, and this is probably the one that have most attention, is the use of cannabis sativa for CBD and THC production. Okay, if you're growing, cannabis sativa to produce CBD, those crop, may not have a high level of THC, and they are called CBD hemp. And most of the attention related to this species mostly associated with the use of this species for illicit and medicinal drugs. so drug type cannabis. Now, one of the interesting aspect about this species, is that there are so many different uses that are associated with this one species. And there are different, in addition to different uses, you also have different legal identity. Like the crop that have a THC level higher than 0.3% dry weight are considered cannabis. They're regulated as Schedule 1 substances. So they are different, uses and identity, and coming with that, it presents a unique challenge for environmental health and research, associated with cannabis sativa, which is the focus that we will be talking about of today's talk. Now, I also want to point out a couple of recent articles. Here on the top, this is a article coming out earlier this year in February in Wall Street Journal talking about cannabis safety. And there's another podcast come out. This just come out 2 weeks ago on New York Times, the Daily. Okay, and this comes shortly after the National Academy report. So cannabis safety is something that had attract a lot of interest, in the media in the past year, particularly in the past year. And you can see like everything that I'm showing here on the screen, this all happened this year. The National Academy report and the New York Times daily podcast, this just come out like 2 weeks ago. So these are a very recent development. And as you will see later in this talk, you will see a lot of similarity in how people approach cannabis safety, like using the methodology from food safety, and there are pros and cons, with this approach, we'll talk about in a bit. Now, looking into a species with multiple use and identity, my lab approached this research problem using the One Health approach. So the One Health approach is an integrated, unifying approach to balance and optimize the health of people, animal, and the ecosystem. So we are not just interested in the public health implication of cannabis and industrial hemp. We are looking into it, in both from both animal health and environmental health perspective. And one of the strength of using this approach is we leverage the close interdependent link among different fields to create new surveillance and disease control methods. So this is the approach that we are taking in the Cannabis One Health Lab at ASU. And the core principle, we try to use different, knowledge research methods from different disciplines to tackle research questions related to cannabis and environmental health. Okay, so this is a summary slide talking about the current Cannabis One Health research at ASU in my lab. Today, we are only going to focus on two aspects of our research program. One that related to the regulatory policy of pesticide, microbe, and mycotoxin and cannabis, and the other focusing on the solution, which is to use C. elegans assay, as I will talk about in a bit, looking into neurotoxicity of cannabinoid and pesticide contaminant. Okay, next, so this is, after an introduction, I want to talk a little bit more about environmental contaminant in cannabis and how they're being regulated right now in the US. Okay, so first, what are environmental contaminants that can be found in cannabis? Now, there are four major categories of environmental contaminants. The first category is pesticide, things like chlorpyrifos, mycobutanol. So what I'm showing on the screen right now are two pest and fungal problems that are very common in cannabis production facility. The creature that I'm showing here on the right-hand side, two-spotted spider mite. This is one of the major pests in cannabis production greenhouse. a lot of the greenhouse that produce cannabis, these are not standard, agricultural biotech level kind of facility. And if one of these, get into your lab coat, like getting into the production facility, you would never get them out. Okay, really, really nasty creature. Tremendous damage can be caused to the crop, and people do use pesticide to fumigate the facility, like with infestation. So the one of the issue why people use pesticide in cannabis production. Powdery mildew is another problem in cannabis production. Can be really devastating because it can happen at the end of the production during of cannabis. It can devastate the cannabis production industry. Like when it happens late. Like in when we're close to harvest of the crop. Now, one issue I want to brought up in this category of contaminant is that up to 90% of the detection rate in illegal cannabis in Canada has been reported in a recent study. This is one thing that I think it deserves more attention, which is not just pesticide, but contaminant in general in illegal cannabis. Because, often when we talk about cannabis contaminant, often we are talking about the contaminant in, legalized cannabis, but illegal cannabis still, have a large market share even in legalized market. And the contaminant is a key concern for public health. So something to keep in mind as well. Next, toxic element. Yes. So obviously, when we talk about toxic element, they include metals element. So lead, cadmium, and mercury. These are some of the contaminants that not only has been reported in cannabis, but also has been reported at a higher level in cannabis user. So there is a report coming out last year talking about cannabis users have a higher serum and urine level of lead and cadmium. And this is the data that are collected before 2018. That is when the cannabis production in the US, a lot of it is still, like in the black market. It can come from the hyperaccumulation of element of the plant. So that would be a natural process. Or it can also come from the packaging process. The metal can leak into cannabinoid oil, like if you have, like heavy metal in the packaging material. Okay, so toxic element is another key concern, another category of contaminant. Also, solvent. Now, this is a category of contaminant that is specific to cannabis. Okay, you have solvent like butane and propylene. This is also, a key concern for illegal cannabis as well. Okay, so how are environmental contaminants being regulated in cannabis in the US? Now, first of all the category of contaminants that I've talked about, there's no federal regulation, because of the Schedule I status of drug type cannabis. And this is different for hemp, because for hemp, after the passage of the 2018 Farm Bill, it is covered by Federal Insecticide, Fungicide, and Rodenticide Act, FIFA. I think many of you in this audience know what it is. But one thing that I want to brought up is that there's no clear enforcement mechanism. Like from what I have heard so far, about intoxication hemp product, right, and also the contaminants that are in it. Okay, and also there is no legalized use of pesticide in cannabis. There's only a couple of active ingredient that have been investigated in hemp, and the US EPA actually have a list. I believe it's I think it's somewhere between 60 to 100 active ingredient. I don't know the latest number, but most of these are not conventional pesticide. A lot of these are low impact pesticide that have FDA guidance on their usage in hemp. So definitely an area, even in hemp, like that need to be developed. Now, how about the regulation at the state level? There are different approaches that are being implemented right now by some 35 states and Washington, D.C. with legalized cannabis that we will talk a little bit about in a bit. Most of the state regulate pesticide use in cannabis, as agricultural and food commodity. This is where they take their reference of regulatory policy from. And also some state interestingly regulated, as an organic food. Okay. So how does the whole landscape of cannabis pesticide regulation look like at the state level? What I'm showing here is a Sankey diagram of the four categories of contaminant: pesticide, solvent, a microbe, and mycotoxin, which I will talk a little bit more at the end, and also inorganic toxic element. Okay, as you can see, this is the data collected in 2022. Okay, if you look into the regulatory document from all the legalized jurisdictions at that time, you can find a total of 679 cannabis contaminants that include 551 pesticide, 64 solvent, 12 inorganic, 24 microbes and mycotoxin. That is the one that, like the smallest group here, as well as 16 other contaminants. Now, the reason why there are so many, of contaminant is because individual states, they set their own list of analytes, for compliance testing. Right? Each state have their own regulatory policy. So individually, one state program can have a list of analytes to look at in compliance testing, between 60 to 120 analytes, okay? But collectively, if you add them all up, nationwide, using the 2022 data, there is over 600 of them. Okay, that just gives you an idea, how, much of a variation, there is, when it comes to regulatory policy in cannabis as the national level. Okay. Now, in this graph diagram, I'm showing the number of analytes for pesticide, solvent, inorganic toxic element, and also microbe. Okay, and on the x-axis here on each panel, I'm showing the number of listed analytes, for compliance testing in each state. And then on the y-axis, I'm showing the number of jurisdictions that regulate the corresponding number of the contaminant, under each category. Okay, I'm going to go through them one by one. So first, oh, sorry, I just saw the message. Am I still having echo? Maybe I'm just speaking too loud. I'm just going to slow down a little bit. Sorry about that. Okay. All right. All right. So pesticide contaminant. So pesticide contaminant, if you look at this list, you can see that there is a number of states, the majority of the state, from, a low number of analytes in the compliance testing all the way to over 100. And then also a number of states that, have a high number, over close to 400, analytes in compliance testing. The reason why is that in this state, they basically take the entire code of federal regulation, the CFR 180, the whole list of tolerance for agricultural commodity, and they say that, okay, whatever on the list that are used for food commodity, we'll look into it. So a lot of them is not relevant to cannabis production. So not entirely relevant to what need to be regulated, in cannabis. Solvent contaminant, there's also a large variation. Some states don't really regulate it. The other state, regulate it over 40 of them. Okay. Inorganic contaminant, on the other hand, is the one that have the most consistency. State that regulate inorganic contaminant, they agree on that arsenic, cadmium, lead, and mercury are the one that need to be regulated. Also, you have states that have regulation of more element. Some states that do not regulate it. Microbial contamination also have very large variation, partly because there's a lot of different microbes, but also there's a lot of different methods to regulate the different microbe, in cannabis. So different method by itself, caused a lot of variation when it comes to microbial contaminant regulation. Using the data, that we collected from the regulatory document in different states, we created a simulation taking a batch of some 9,000 sample from a cannabis testing lab in California and run it through all the regulation in different states. And then we see there's a variation in the percentage of contaminated cannabis that are being caught in each state using those regulatory action level and the analyte for the compliant testing. Right? There's a range of contaminated flower that would not get caught, due to the differences in action level and mostly, analyte that are listed in compliant testing. The point that I'm trying to show here is that this is a unique situation when it comes to agricultural commodity. Okay, so what this diagram is showing here is something that you would not expect if you are getting, say you're getting a apple from a supermarket, right? Like you would expect that, okay, it's the same, action level for the same commodity, produced with, mostly the same production method and guideline nationally. But in cannabis, it's not the same, right? State by state, there are different action level. That's just to show that, this is, an area that, when it comes to research, regulatory research, I mean, there's a lot of, a room for refinement, Okay. Last but not least, I want to show one more figure. This is a figure that, we generated using the data from the report of ADHS, the 2019 document. Back then, Steve and Mary from the state lab, they conduct a comprehensive analysis of the regulatory action level back then in 2019, right when the time that Arizona is having legalized recreational cannabis use. Why when the time that the state the act is passed to legalize recreational uses. Using that data and compared to the data that we collected from the regulatory document last year in 2023, we compare the number of pyrethroid and organophosphate that are regulated, in the US, right? Again, same diagram. X-axis is different number of pesticides that are regulated in each state, and then the Y-axis is the number of, state level jurisdictions that are regulating those number of pesticides. And you can see, the red is the data from 2019. The green is the data from 2023. So you can see that for both classes of pesticide, pyrethroid pesticide, organophosphate pesticide, there is a white shift, right? If you look at the red, in 2019, the median is 0, right? Most of the states in 2019, they actually do not, regulate these two classes of pesticide for some reason. But then, in 5 years later, you see that there is a shift, right? Most of the state regulate more than 4 pyrethroid pesticide and 7 organophosphate pesticide. And as I will show you in a bit, there are a potential mixture effect of cannabinoid and organophosphate pesticide. So this is an area that again there's a lot of changes like with the cannabis industry that are operating in many states now. Okay, so the third part I want to talk a little bit about the health risk to neurological patient and what does it mean like from a clinical perspective. All right. So in this study, we specifically focus on Parkinson's disease patient. This is a clinical population that have a, like many clinical population, have a higher rate of cannabis use at the population level. Between 25 to 40% of the patient with Parkinson's disease have reported to use cannabis as compared to the 80% national average for cannabis use. One of the key concerns that our lab has, given the fact that, like there's some the cannabis safety regulation, this is still pretty much an area of, pretty much an evolving area, is that there is a lack of research and guidance on how to communicate risk of cannabis in a clinical setting, right? That is not just for Parkinson's disease, across the board, cancer, epilepsy, Parkinson's disease, there's a lack of peer-reviewed literature to talk about how to communicate the risk, at a clinical setting. And also in some of the surveys that have been done in the past few years, many patients report that they got little or no guidance, from a patient on how to use cannabis, right? So this is not an area of topic, like cannabis use in general is not an area that people talk a lot at the clinical setting. For that, we set out this study to look into the accessibility and guidance of cannabis use in Parkinson's disease patient. And in this study, we basically do two things. One is we review the state list of qualifying conditions just to see what medical condition each state a qualifying patient for using cannabis, in 2019 versus 2023. And then we also survey patients and basically ask them about their knowledge and risk perception of cannabis use, in Parkinson's disease. Now, in this study, we work with two collaborators. One of them is Dr. Holy Shield in the Muhammad Ali Parkinson's Disease Center at Barrow Neurological Institution. And what we have done with Dr. Shield is that we look into the state qualifying condition document. And then we pick out the conditions that are relevant to Parkinson's disease. There are a couple of them. Parkinson's disease, of course, muscle spasm is another that's relevant to Parkinson's disease patient. Also pain, anxiety, insomnia. Okay, so these are all the conditions that Parkinson's disease patient, first of all, these are for symptom management. Second of all, these are also conditions that are listed, in the state guidance document in different states that qualify them for getting a medical card to use medical cannabis in each state. All right. And also, we work with a public health attorney, Matt Weinsburn, at the University of Maryland, Francis King Carey School of Law. And we collected two sets of data, and these are the data, for the state guidance on medical cannabis use, from 2019 and 2025. Okay, so this is the this panel show the result that we got. We have two rows here, 2019 and 2023, and then we have three columns. The column on the left right here in green is legality of cannabis use, in different states. Okay, so you can see the lighter shade of green is medical use. And then the states that have legalized medical and recreational use are in dark green. Okay. And also you have Parkinson's disease, the state that explicitly list Parkinson's disease, as a qualifying condition is in blue. And the last one is pink, like that is that are visualized in pink color. So what I'm trying to show in this diagram is that if you look at, the legality of cannabis use, right? First look at the column on the left. You can see that across the board in the past 5 years, there are more states, like having legalized cannabis program. And this is in parallel with the increased public perception, of cannabis use in general, right? So that is well known. But if you compare them, with the medical use, you can see that not only that there are more states that have cannabis program, but also if you look at individual state, right? So you have clinical population that are getting access, to medical cannabis, then the number is increasing. Pain is a group that is a condition that are particularly have I would say this is the group that probably have number one, they're more studied too, and also this is the group that most states, as you can see in the color, are listing pain as a qualifying condition, like across the board. Now, so with what I have just showed you before in the previous slide, there are more assets, like for cannabis, including a clinical population for a patient with different medical condition. What do patients think about the risk of cannabis contaminant in patient with Parkinson's disease? Now, this is a very specific question. We're very interested to see how much physician know about it. So we collaborate with Dr. Shiu in Barrow Neurological Institute, and we conduct an online survey of neurologists and movement specialists. Now, these are a board certified medical profession, okay? And this survey is conducted between July 27, 2022 and June 15, 2023. And the question that we asked this group of physicians focus on physician practice information, how many patients are they seeing, and also the cannabis program in their state. Do they know much about the cannabis program in the state that they're practicing in? We then ask them about their perception of risk when it comes to cannabis and also cannabis contaminant for the Parkinson's disease patient population. Last but not least, we also ask them their opinion on how to improve cannabis use safety. Okay, so in the first set of questions, when we asked, the clinician, again, neurologist, movement disorder specialists, right? So what do they think about the risk of cannabis contaminant in patients with Parkinson's disease? Now, this is a very interesting, breakdown here. We have close to half of the physicians saying that the risk is unknown. Okay, there are physicians saying that they think that this is no risk. Some physicians say it's moderate risk, but for the most part, most of the about half of the physicians coming back, they say they just simply don't know what the risk is. Okay, I think it is a very interesting observation here. Now we asked a couple more questions, right? Are you aware of some or all of the qualifying conditions in your primary practice state? Okay, so in general, 4 in 5 of the physician interview, they actually do know what is being listed as a qualifying condition and what is not. But when it comes to the knowledge beside regarding the composition of the cannabis that people are using, that the patient might get exposed to, right? Besides THC, CBD, cannabinoid, and terpene, right? Are there any substances in cannabis product? Like for those of you who are in cannabis safety, you probably will think, okay, this is kind of a trick question, and it is because this is a contaminant question. And you can see that a lot of the physicians, off the top of their head, they actually don't know about the impurity, right? Out of the 20 responded, the majority of them say, I don't know. Now, going from there, so what did we do? So basically what we did is we show this physician, this table that I'm showing on the screen. Okay, and this is the table that my lab put together. This has the basic fact when it comes to cannabis contaminant that I just talked about earlier, like in this talk, right? Like, what are the contaminants? Where are they coming from, right? This is a factual, a table just to it basically just give you the fact about cannabis contaminant. And after we give the table to the physician and we ask them for the opinion again, boom. Okay, so this is the opinion that we collected, regarding their perception of the health impact of cannabis-borne contaminant when it comes to Parkinson's disease. A lot of physicians say comorbidity is an issue. Long-term effects is another issue. Potential policy changes to reduce health risk of cannabis-borne contaminant. A lot of people say, okay, we need to regulate this. Like some say, we do need to better educate the healthcare provider. The take home from this diagram is that, when we are talking about cannabis safety and when we are talking about it in like how we engage healthcare provider, I think to be able to transfer the knowledge, basic knowledge and fact, right, to the clinician is key. It doesn't have to be a lot, right? Because this is not a lot of information. But after we give this information to the physician, now remember, these are physicians that are experienced in working with Parkinson's disease patients, right? They are highly educated healthcare provider. Like they actually have a lot to offer, right? To be able to transfer that knowledge of, okay, this this contaminant can be found in cannabis, that immediately empower them to provide their own clinical opinion on the safety issue when it comes to cannabis. So I think this is something that we learn, like when we come to educational intervention, like just to be able to provide the basic fact, like to the healthcare provider is key. Okay, so I want to switch to the next topic and talk about solution. Okay, like given all these contaminants, okay, and then it can be used in different for different conditions, right? So it's how do we approach it? Like from a toxicology point of view, like how do we get the toxicity information? So when it comes to toxicity assessment of cannabis contaminant, this is a very challenging subject. Like first of all, we're dealing with one commodity, cannabis sativa, and drug type cannabis sativa, right? But it has a variety of contaminant that can be found in it. And when you talk about the cannabis sativa and drug type cannabis in general, you have the cannabinoid, and you also have the other contaminant. There might be toxicological interaction, right? So how do we address that complexity, right? You have patient, susceptibility to the health condition, right? Some patient might be more, susceptible to a certain group of contaminant than the other. Also, keep in mind that this is an area that are rapidly evolving, right? Like right now it's at the state level. Don't know how the federal regulation is going to change, like at this point, I guess. Overall, it's a rapidly evolving regulatory landscape, right? So how do we keep up? Right? How do the toxicology keep up? One of the solution is the new approach method. So new approach method is a group of alternative method of weapon toxicity assessment. Okay, and different from the traditional description approach relying on conventional mammalian model. Like this method is there to predict potential health risk, right? To basically get mechanistic information from the mechanistic information first to guide further laboratory study, rather than just, okay, let's just first start with a model, right? So it's a different approach, and I think that they have a lot of opportunity to using this kind of method. And the two methods that we have used, under this new approach methodology, first is bioinformatics. So we use a database called comparative toxicogenomic database. So this is a database that is based on literature. It allows us to generate the information at gene level. I'm going to show you a bit how to use it. The second method is to use a new model organism called C. elegans. Okay, so I am one of the earliest scientists to develop this model. Has been a while now, as you can see. I'm going to show you all how to use both methods to do toxicity testing with cannabis pesticide mixture. So comparative toxicogenomic database is a curated literature database that capture peer-reviewed open scientific literature, and it captures 4 components of information: chemical, gene, phenotype, and disease. Okay, and basically what we are doing here is using this literature database. Like we look across all the information in this database, and we basically ask a hypothetical question, right? If you have Parkinson's disease patient, they're exposed to contaminated cannabis, what are the key gene level target that both the cannabinoid and the pesticide is going to hit? Okay, so that's the question, right? So after that, like what we have done is that we look into the database and we ask, how many contaminant and cannabinoid can we find in this database? So now on the left-hand side here, these are the chemical, and then on the right-hand side are the gene. Okay, and the line here indicate the number of connection that the database can find based on the comprehensive literature database that the literature, the article that the database pull from the open literature, like PubMed. Okay, so basically, if you look at, now this is pretty small, but if you look on the left-hand side, basically what we find is the green boxes here indicate the cannabinoid that are in the database, the 15 of them. The orange here is the pyrethroid, okay, in the database, 10 of them. And then a lot of the organophosphate, 27 organophosphate can be found in this database too. And a lot of these pyrethroid and organophosphate, a lot of these actually are the one that, remember in the earlier diagram, I showed that, there are state regulating these 2 chemical class of pesticide. A lot of them are listed by those states, actually. So this is definitely a useful resources. And then on the right-hand side, I'm showing that the boxes that have the largest size indicate the gene target that have the most literature support. The top one being the tumor necrosis factor, like followed by interleukin-6, superoxide dismutase 1 and 2. So this gene, for those of you who are familiar with functional genomics, these are targets that point to oxidative stress. So this gives us some idea of how, when you have a situation that the cannabinoid and pesticide, they both hit the new one, what mechanism would the cell be hit with, right? So the predictor mechanism is oxidative stress in this case. Next, what we do is test the C. elegans model that I just showed in the previous slide, the alternative animal model, with 4 chemical, copyrofos and organophosphate, okay, permethrin, that pyrethroid. Both of them has been reported, like in both legal and illegal cannabis, okay. And then you have CBD and delta-9 THC, right? So the x-axis here is showing the dose of each chemical. The y-axis is showing the repulsion time. So that is a behavioral, basically how the C. elegans retract from a stimulus. And that is a reaction specific to dopaminergic signaling. So we basically look into this, like particular signaling, susceptible to oxidative stress, like relevant to Parkinson's disease pathology. We see how each chemical individually, how it hit the C. elegans model with this mechanism. And as you can see, in this diagram, the cannabinoid itself, there's no dose-dependent effect, right, on the right-hand side, which is not surprising. I mean, after all, we are talking about recreational substances, right? We don't expect for this kind of assay it would show high toxicity. That would be surprising. Now, pesticide, on the other hand, like two different pesticides, you can see that different dose respond. The copyrifos showing really strong response here. Okay. So next, what we do is we mix copyrifos, the organophosphate, with the two cannabinoids, and that's what we got. Okay. Now again, like the x-axis are different doses of copyrifos, right? But this time they're mixed with different cannabinoids. And then on the x-axis, same, you have the repulsion time, which is a readout, for dopaminergic signaling, how it's being attacked by the mixture of chemical. What you can see is first, it doesn't show the synergistic effect that we expected. Doesn't seem like you add the two together, it's going to have an exponential large effects. Like it's not. We don't have that. But what we have instead is differences in the dose response of copyrifos in the presence of cannabinoid. Because if you look at the previous slide, the cannabinoid by itself, they do not have a detectable effect. But when you add it together, then the copyrifos is having a different effect. So one example that I want to bring up here is, you think about regulating copyrifos in the watermelon, right? You worry about the copyrifos toxicity in the watermelon. But then if you're talking about cannabis, there might be interaction. The toxicity of the copyrifos might be different, right? So there are some, like difficulty when you're making that human health hazard assessment, like knowing that there's interaction, the food component that you're putting the pesticide in is affect the toxicity of the component by itself. And there's a mechanistic reason for it. This is a diagram showing the three different components of the endocannabinoid signaling pathway. You have the G protein coupled receptor pathway that respond to not just delta 9, but also the endogenous cannabinoid. You have the ligand gate channel pathway. So this is how the synapse, communicate, right? So the green one is the retrograde signaling. The yellow one is the one that to go, like from the P synapse to the post synapse. You've got two different pathways that are in action here. So what about the organophosphate? How does that affect the signaling? It hit it here. Okay. So what I'm trying to show here is that there is a Q switch for the endocannabinoid that are in the neuron, right? So the THC, they will add to it, but the neuron itself have endocannabinoid, and there's a Q switch, the serine hydrolysate. And the Q switch itself can be inhibited by the organophosphate pesticide. So there are potential mechanistic interaction in it, right? So just to show the opportunity of using this kind of new approach methodology, we can actually get to the toxicological interaction in a mechanistic level, right? Which is actually pretty hard to do. We just go to study the chemical mixture with rodent model, like the conventional method. I have about 9 minutes. I'm going to wrap up by talking about a new research direction. Just a couple of slides here that my lab is undertaking with fungal and mycotoxin contaminant in cannabis. Now, cannabis, like other agricultural commodity, it interacts with a community of microbes. So in this paper that I'm showing here on the screen, I'm reporting, with my collaborator from Tennessee and Canada, the mycotoxin and also fungal contaminant that can be found in cannabis and hemp. There are over 100 of different fungal species in cannabis and hemp. Some of them can produce secondary infection, other can produce mycotoxin. Now, if you're looking into, again, the diagram that I showed earlier, the big picture of the regulatory landscape for cannabis contaminant, one of the things that you will notice is that there is a lot of interest in pesticide and solvent. A relatively less attention is given to the regulation of microbe. Like I'm talking about looking into what is in the cannabis, like when it comes to different kind of microbe and mycotoxin and what is not regulated, right? So it's the regulation fit with what are the analytes that may cause problem in human health. And if you look closer at mycotoxin, one of the things that you will see is that there are only 5 mycotoxins currently being regulated in legal cannabis in all states and all countries that we know of. Aflatoxin B1, B2, G1, G2, and ochratoxin. And a lot of these regulation, when it first set in place, these are coming from food safety regulation, which basically they take the action level and they see, like what should that be, in cannabis, without looking into what mycotoxin is there, right? At that time, the information was not there, and there's still an information gap. Specifically, the regulation focus on Aspergillus species, as you can see, the 5 that I listed here. And there is no country or any state in the US right now currently require any testing for the presence of Fusarium, which is, a group of mycotoxin that we think might get overlooked. And also, when we're talking about all the cannabis used in the US, black market remain, to be over 1/3 of the cannabis market in the US, according to the latest statistic. Okay, and you have, mycotoxin that, like have not been tested, like in the legal market, and then also there also little information on the black market cannabis, like and in some states that's the only sources of cannabis that is available. There are several challenges when it comes to mycotoxin assessment in cannabis. One of the challenge is the regional specificity when it comes to the prevalence of mycotoxin, right? To study mycotoxin, you really need to have a nationwide study, right? And then because of the legality of drug type cannabis, I mean, to do those two studies are very, very, very difficult. And also there is no toxicity information for Fusarium mycotoxin when it comes to inhaled cannabis. Okay, not that there's no information on inhaled Fusarium mycotoxin, right? Like for those of you in environmental health, you know about like 6 building syndrome. So that is, like associated with Fusarium. But when you're talking about, like Fusarium that might come into vape pen, like that is, or like other form of cannabis, that risk assessment is not there yet. That's what I'm trying to say here. Now, vaping device in particular, there's a FDA study showing that the vaping device is not powerful enough, like to kill microbe. And also the Fusarium mycotoxin, like to cannabis consumer, that connection to adverse health effects still need to be worked out. But I can report that the vomiting syndrome, you guys might have heard about cannabis hyperemesis, like is a really is a topic that have gained more attention, in the media as well. It have similar vomiting symptom with the Fusarium mycotoxin poisoning. Okay, so with that, like I would like to close. First of all, environmental contaminants, like pose a health risk to cannabis user, and the cannabis safety information that has to be an important part, when it comes to physician and patient education, about cannabis use. A new approach method, such as the bioinformatic and C-elegant model that I show you all, provide a new solution to advance some of the knowledge gap that we have, like when it comes to cannabis contaminant mixture. And we need to have more research to address the risk of fungal and mycotoxin contaminant. With that, I would like to end my presentation with an acknowledgement of all my collaborators and funding support. Special thanks to the Department of Health Services, which allow us to do a lot of our education and outreach. So Travis talked about the seminar, a moment ago, and in addition to the seminar that we run every semester, next year we also have a Cannabis Sativa testing workshop at ASU. So stay tuned, and thank you so much for your interest and time.
Thank you, Max. That was very, very interesting. I've definitely learned a lot about the whole marijuana field since I've actually been working for the department. I actually started in 2021 as the marijuana kitchens inspector, and it has been incredibly helpful. And I would definitely highly recommend any of the workshops that ASU and Max and his team do have. I've had the privilege of actually going to a few of them. The last few that I know that they've had in person and virtual options. Definitely a wide range of subjects in regards to cannabis. So if you do have the opportunity to attend those, I would highly suggest those. So it looks like we have like a minute or so just to do some questions. So we can go through those, and then we will be taking just a short break. So it looks like as of right now, we do have 2 questions. So if you do have any others, just please put them in Slido. So it looks like the first one is, does the health impacts of contaminated cannabis vary with method of delivery via, like say smoking, inhalation, or edibles?
Yes. So regarding the method of the delivery, at least for the cannabis used for medical purposes, according to Health Canada's guideline, it generally not recommend smoking as a method. The reason why is, if the patient is sick, it might be immunocompromised, and if you're taking the cannabis in smoke, which is not sterile, that it can increase the risk of secondary infection. So that's one. And the second question about cases of Aspergillus infection from cannabis. So there are some. So in this, like we actually did a comprehensive literature review on that question on that question in our paper. The short answer is there are some really old paper that look into it. But recently, and there's one recent CDC paper that used health insurance data to build an association between fungal infection and cannabis use. But this is not an area that has a lot of research on, like partly due to the difficulty to obtain federal funding, right? But you think about asking questions like this. This is not a cheap question to get an answer. So we definitely have a knowledge gap to fill, when it comes to cannabis safety.
All right. Well, yeah, thank you. And I mean, just to add just a small little blurb to that, since recreational marijuana has been legalized, we have done several, at least at the state, we have done several marijuana recalls in regards to Aspergillus. As of today, I believe that there has been one potential suspected case, but that has not been confirmed. So as our knowledge is, so far, there's one suspected, but nothing yet confirmed. So just to add on to that. So thank you very much, Max.